Thank you so much. Good morning. It's my pleasure to be here to provide a little bit of an overview for session 5, an overview of the current science-based regulatory standards for topical and transdermal drug products. Now, topical drug products, of course, are those that are locally acting. So these are products that are applied to the skin and intended to act locally, typically in the skin. Typically, these are dermatological drug products. Transdermal drug products, thank you, are those that are applied on the skin but are intended to act systemically. So these are often the fentanyl or nicotine type products. What I'd like to be able to talk about briefly, as providing context, is that we understand these are complex and challenging products, and the challenging product development issues have represented a hurdle, again, have provided a barrier to the development of generics. Now, the next several speakers during session 5 will discuss some of the scientific considerations that contribute to the complexity of these products. They'll describe how research funded by GDUFA, performed within the agency and in collaboration with researchers across the world, have helped to overcome these hurdles and to reduce the barriers to generic drug development for these products. Now, this has led to the development of more efficient, science-based regulatory standards. This morning's presentations are not just an exercise in storytelling, though, or in celebrating the successes of the research. This is a workshop. And so the material is intended to help you understand the key insights that we've learned from the research, but how to ensure that your prospective generic products perform the same despite inherent complexities, so that you can develop high-quality generics, so that you can avoid common mistakes and common deficiencies during the review process. And we're happy to discuss issues specific to your product during development through control correspondences and through the pre-ANDA meetings process, because we want to facilitate not only approvals, but first cycle approval so that patients can have access to high-quality, complex, generic topical and transdermal products. So with that, let me move forward to also say that this presentation reflects my views and not the official positions of the agency. So when we talk about topical and transdermal products, their reference listed drugs are often complex in multiple ways. And there's some unique considerations that impact equivalence. So what I'd like to do is briefly discuss them independently. First, we'll touch on topicals and then on transdermals. So we know for topical products that the formulation matters greatly. We've known this for a very long time. But increasingly, through the research that we've done recently, we begin to understand why the formulation matters so greatly. It's because the components and the composition, the components being the ingredients, and the composition really being the formulation, they modulate the physical and the structural arrangement of matter. And the resulting topical product characteristics can influence the metamorphosis and bioavailability. They influence that rate and extent to which the drug becomes available at the site of action. So how does this work? Well, the components and composition and the physical and structural properties influence the drug states and the phases of the dosage form. Is the drug dissolved? Is it in crystalline form? Is this a solution? Is it an emulsion? What are the phases of the drug product and of the dosage form? And Dr. Ghosh will be talking about this in a little bit more detail. Now, if you have multiple phases in the dosage form, this becomes a little bit more complex, because then the question is, where is the drug distributed in these phases? If, for example, the drug is dissolved in globules, well, then we have to consider the diffusion of the drug within the globule, the partitioning from the globule into the continuous phase, which would occur across the surface area of the globule, which is a function of the globule size distribution. It then has to diffuse through the continuous phase and partition from the formulation into the skin at the stratum corneum. But it's not just diffusing from the continuous phase. You're going to have globules that are occasionally impacted down upon the skin, and it's also diffusing directly from the globules into the stratum corneum. And it's not just the drug that's diffusing. The components, the ingredients of the vehicle are also diffusing, and they're changing the solubility of the drug in the skin. They're altering the structured arrangement of matter in the skin and changing diffusion. And so all of these processes are described a little bit on this slide, and ultimately this affects the drug delivery and the bioavailability of the target site. One of the other things is that the physical properties of the vehicle can affect the skin hydration or dehydration. Is it an alcohol-containing formulation? Is it aqueous? Is it an occlusive ointment? And these things can also affect how irritating or damaging to the skin the formulation may be. What it also affects is the metamorphosis of the dosage form on the skin. Our research has conveyed recently how powerful this effect of metamorphosis is, that it's not just about the dosage form at the time when it impacts the skin, but actually as it dries, as it undergoes metamorphosis throughout its time on the skin, because that drying leads to changes in the composition of the vehicle. Certain components of the vehicle will potentially evaporate away. This changes the vehicle, this changes the thermodynamics of the drug in the vehicle, and these things all affect the bioavailability. So how do we control these various factors of components and composition and arrangement of matter? Well, we have concepts related to Q1, Q2, and Q3, where when a generic product is Q1 the same, it has the same ingredients, the same components. Q2 sameness, of course, the same components in the same composition, the same formulation. And Q3 is the same components, the same formulation. So it's Q1 and Q2, but it's also the same arrangement of matter. Now we say similar here because just like for Q2 sameness, there's a plus or minus 5% tolerance there. Similarly, for different batches of the reference product, these physical properties may exist within a range. And so when we talk about that Q3 similarity, we're really talking about Q3 sameness within the range of the characterized variation for the reference product. So what does it do to us to be able to control Q1, Q2, Q3 sameness? Well, when we're able to control Q1 and Q2 sameness within a topical generic, this mitigates the risk of some known failure modes related to irritation and sensitization, for example, or the way that the formulation interacts with diseased skin, any differences that might arise otherwise from stability or solubility of the drug. And also, this is particularly important for topical products because the vehicle itself may have a large contribution to efficacy. Its physical features, its occlusivity, for example, may be very important to the efficacy that arises from the topical product. And so we can build in bioequivalence, sort of a bioequivalence by design concept when we match these characteristics of the reference product. And then there's also the question of the arrangement of matter. Beyond the components and composition, which can have a certain tolerance in them, you could have potential failure modes that are related to differences that may arise from differences in pH that may sting or irritate diseased skin, or differences in the polymorphic form of the drug, or in rheology, and in the air that may be whipped into the product, differences in phase states or diffusion or partitioning, and particularly differences in the metamorphosis. We've actually seen products that are Q1, Q2 the same, this has been part of our research, that if you change the manufacturing conditions of Q1, Q2 identical products, you can actually alter the water activity and you can alter the drying rate and alter the bioavailability of the drug from these formulations potentially. So the Q3 is a very important aspect of being able to build in that bioequivalence by design. This then leads us to a modular framework for in vitro BE, where you kind of have a bioequivalence by design type of approach, where if the generic product is Q1, Q2 the same, Q3 similar, then we've systematically mitigated the risk of several failure modes for bioequivalence. One of the challenges is that as the dosage form becomes more complex, it's challenging to know. We're still building confidence about whether the Q3 characterizations that we recommend are sufficient to characterize all the potentially critical quality attributes. And that's where also including an in vitro release test, a performance test, can help to implicitly tell us something about other differences that could potentially exist in the arrangement of matter that we may not have characterized. And as the formulation becomes more complex, like into emulsions, to have a performance test like the in vitro permeation test that allows us to actually evaluate the cutaneous pharmacokinetics in vivo to really get a biorelevant evaluation of the performance of the dosage form. Now, in addition to this, there may be some topical products for which the site of action may be both locally and there may also be a systemic contribution to the local effect. And therefore, there are multiple approaches that sometimes we also have in vivo PK studies that might contribute to the mechanism of action for a topical drug product, and that may be also something that can be built into the paradigm of the collective weight of evidence. Or independently, there may be pharmacodynamic vasoconstrictor studies that are, that are relevant for topical corticosteroids. Clinical endpoint studies are widely amenable to many topical products, but they're challenging to conduct and perhaps not the most sensitive. One of the things, though, that we'll be talking about this morning, Dr. Eleftheria Tsakalozu will talk about some of the quantitative methods, the modeling and simulation that I strongly encourage you to think about as part of that collective weight of evidence. Some of these products are very complex. There's only so much empirical evidence that can speak to bioequivalence, that can speak to mitigating the risk of failure modes, and that's where involving these in silico methods will be very, very important. Let me also speak very briefly about transdermal systems. I've got depicted here on the screen in the top panel, that's a reservoir system, sort of the old bag of gel type of transdermal system. Most of the modern systems are matrix TDS systems, although there's quite a bit of variation even among them. I'd like to illustrate here that there are design differences where even if the reference product was a was a reservoir product, it's certainly allowable and encouraged for the generic to be developed as a matrix TDS. However, because there can be differences in the product design between the reference and the generic product, there can also be differences in the potential failure modes associated with those products, and that's why product characterization using a variety of different approaches is very important. For example, compared to the reference product, the TDS, the generic TDS may have a different drug load and a different residual drug excess. And in fact, we encourage that it should have a lower or comparable drug load or residual drug excess. Similarly, it may be different in product size and shape. Hopefully, you would not want to develop a generic product that's a lot larger because the patient may not find that very acceptable. But we'll have some talks. Dr. Ramazanli will speak about some of the implications of size or shape. There are some implications for product strength when you have different sizes or shapes, and she'll speak a little bit about proportionality. And heat effects is something we're particularly interested in. We've learned through our research over the last several years that in a manner analogous to the way that many solid oral dosage forms experience dose dumping when they encounter alcohol, the analogy for topical drug products is when they encounter heat, that rate of drug delivery may change. And because of formulation differences or design differences with the generic, you want to ensure that those heat effects are no worse for the generic product. And there's been some research that has led to the development of some efficient in vitro approaches during product development that can evaluate that. Keep in mind also that differences in formulation, meaning differences in some of the inactive, quote unquote, inactive ingredients, even though these inactive ingredients may be functional like adhesives, and the adhesives may contain impurities. And when you think about the inactive ingredient limits there, keep in mind that this is now in a different formulation that may be for chronic use rather than acute use, that the new formulation may have penetration enhancers. And so be mindful of how these differences in formulation might be relevant for your particular drug product, how they may have a different level of exposure to adhesive impurities, how there could be differences in irritation or sensitization potential, or adhesion characteristics, or heat effects. And ultimately, we must make sure that these products behave the same for patients. And that's why when we look at the in vivo studies that are part of the collective panel of studies to demonstrate bioequivalence for a transdermal product, we think about the following three studies. First, an in vivo BE study with pharmacokinetic endpoints. Second, a comparative study evaluating the adhesion performance of the prospective generic and the reference TDS. And a comparative study comparing the irritation sensitization potential of these products. So while there can be these allowable differences, we do these studies so that we ensure that it'll be the same for the patient. Now, in addition to these in vivo studies, one of the, I think, really valuable things, and Dr. Ramazanli will talk a little bit about the research and the data that have supported the development of this through GDUFA, are the use of IVPT studies, an in vitro study to compare the effects of heat between reference and test products. And these are actually reviewed as part of the quality of the drug product. So I want to acknowledge a great number of people, so many, in fact, that it would be hard to draw the line as to who would include and who I wouldn't. So I mentioned 4 people in particular here who every single day have day in and day out just been incredible champions that have moved this research forward. Let me also say that we're going to have another workshop in November at AAPS that beyond just understanding what you can do during product development, this will be a real experiential opportunity, the hands-on workshop of going through, kind of like being in a flight simulator, of going through the development process, thinking about these scientific considerations and about actually understanding what our pre-anda process is like, when you can ask kinds of questions, how you ask them, how you frame things. I strongly encourage you. We're putting a lot of effort into giving you the experience to be able to really interact with us very efficiently. So with that, I'll say thank you. I hope I've provided good context for the talks that will come up during the session. Thank you very much.